Hello, everyone. Thank you for joining us for part two of the HCA's first webinar on coffee leaf rust. Our guests today are Kevin Hoffman and Lisa Keith. Our focus today is on regulations and research as relate to coffee leaf rust. For those that don't know, the Hawaii Coffee Association has been hosting weekly conference calls that have been very well attended across multi-agencies, including USDA, HDOA, PBARC, HARC, industry stakeholders to address this issue. My name is Chris Manfredi, and I serve as the president of the Hawaii Coffee Association. Our first guest today is Dr. Kevin Hoffman. He's the administrator of the Hawaii Department of Agriculture's Plant Industry Division. The division is composed of three branches: plant pest control, plant quarantine, and pesticides. The programs of the division are designed to prevent the entry and spread of harmful plant pests, to reduce the negative effects of established pests, and to regulate the sale and use of pesticides in Hawaii. Dr. Hoffman has received a Bachelor of Science degree in Biology and Chemistry from Bowling Green State University and a master's degree in entomology from Ohio State University. He earned his PhD in entomology from Clemson University in 1992. He has authored 21 reviewed publications on insect identification, biology, and invasive species management. He worked with the California Department of Food and Agriculture Pest Detection Emergency Projects Branch for 24 years before joining the Hawaii Department of Agriculture in 2019. So please join me in welcoming Dr. Kevin Hoffman from HDOA. Kevin?
Good morning, everybody. I'm going to go ahead and bring up my presentation. And thank you for that introduction, Chris.
Thank you for joining us.
Okay, can everyone see it? I think, okay. Today I'm going to talk about the Hawaii Department of Agriculture response to the discovery of coffee leaf rust. So to give you some background, on December 21st, 2020, leaf samples displaying coffee leaf rust symptoms from managed coffee plants in the Haiku area of Maui were turned into HDOA. They were based on the physical characteristics and the host expression. Staff were attentively identified the pathogen as coffee leaf rust. Samples were submitted to the University of Hawaii, College of Tropical Agriculture and Human Resources, CTAR, and USDA National Identification Services for identification confirmation. On October 26th, CTAR tentatively identified the pathogen as coffee leaf rust. 2 days later, on October 28th, USDA and NIS notified HDOA that the identification was confirmed as coffee leaf rust, Hemilea vastatrix. This is important because coffee leaf rust is a USDA regulated pest, and so they have the final word on identifications and confirmations and so forth. And I'm sure you saw some this morning, and you'll see some of it in the presentation after me, but coffee leaf rust symptoms, orange, it's kind of orange to yellowish orange in color, spots on the leaves. On the undersides leaves, you'll get powdery spores, which is something that is unlike you'll find on things like Cercospora that's already here. Based on the identifications, subsequent surveys on Maui found plants with symptoms at additional locations, including wild coffee. On October 31st, UHCTAR staff received a sample of coffee leaf rust collected by a grower on a farm on the west side of Hawaii Island in the Kona area. The sample was submitted to USDA NIS, and HDOA was notified on November 10th that it was confirmed as coffee leaf rust. Two samples from the east side of Hawaii Island in the Hilo area were also sent to USDA NIS for identification at the same time, but these came back as negative. HDOA has continued to survey on Maui and Hawaii Island to determine the extent of the infestations. Surveys are ongoing on Kauai and Oahu, which have to date both been negative for coffee leaf rust. Partners on both Lanai and Molokai islands have been informed and asked to look for the disease, and to date, we've had no reports of CLR symptoms. Coffee leaf rust impact is a serious disease of coffee. Causes severe leaf drop, as well as a severe reduction in yield if untreated. This picture here is wild coffee on Maui. You can see pretty much it's just a stick in the ground at this point, with pretty much been totally defoliated from the disease. This is a map of Maui showing the survey results as of last Friday, and so the two blue spots are the major coffee growing areas. The green pins are areas where we surveyed and did not find coffee leaf rust, and the red spots are areas where we surveyed and found coffee leaf rust or presumptive coffee leaf rust symptoms. And the size of those pins for the coffee leaf rust finds are kind of roughly in proportion to the intensity of the infestation. You can see it's mostly northeast side of the island, although there was one find down in the southwest. It's a map showing survey efforts on Hawaii Island, again, as of last Friday. The small black squares are sites where we looked and did not find it. The red dots are where we looked and found either positive samples or presumptive positive, meaning the symptoms were pretty typical for coffee leaf rust. And so we have a cluster of finds in the Hilo area, and then on the other side of the island in the south, and then along in the Kona area on the west side. Next is a map of Oahu, and all the squares here are sites where we looked and did not find coffee leaf rust. And again, we've not found any samples or presumptive positives on Oahu. On Kauai, again, there's 6 areas indicated here as black squares where we looked and did not find coffee leaf rust. And surveys on all these islands, as I said before, are continuing. Kind of what we've seen on Maui is that there's certain climatic conditions that seem to favor large outbreaks of the disease. One is water, for example, rain or mist or overhead irrigation, and splashing water. High humidity, most severe symptoms have been found in kind of a narrow elevational range of 300 to 1,200 feet. Temperatures, daytime between 59 and 85 degrees Fahrenheit, partial shade, and shelter from the wind. These conditions all seem to favor expression of the symptoms of the rust. Industry outreach. HDOA on October 23rd notified the industry of the suspect sample from Maui. At the same time, HDOA asked that the industry on Maui not transport coffee plants, coffee green waste, coffee cherries, pulp, used coffee processing and harvesting equipment, used coffee bags or green coffee beans inter-island, and if possible, within the island until more was known about the extent of the infestation and the actual identification of the fungus. On October 29th, HDOA notified the industry of the confirmed sample from Maui, and on November 10th, they notified the coffee industry of the confirmed sample from the Kona area on Hawaii Island. The department has put out a field guide. It has several pages of pictures showing what to look for if you're out in the field looking for the disease. And that's available on the HDO website as well as a couple of other sites. And at the back of that field guide, there's a a sample form that kind of gives instructions on what to do if you think you see coffee leaf rust. Taking a picture, recording the information, sending it into HDOA so we can have a look at it. We're at this point, because we don't want to inadvertently have the disease spread by samples kind of moving around a lot. We're asking first for photos so we can analyze them, and then if it's looks like it's a good suspect, then we'll arrange to go pick up samples. Regulatory action, kind of following up on that request of October 23rd to refrain from moving articles which could be infested with coffee leaf rust, inter-island from Maui. On November 23rd, the Advisory Committee on Plants and Animals voted to recommend approval of an interim rule that restricts the inter-island movement of potentially infested articles from the Maui and Hawaii Island, and actually any subsequent islands that are found to be infested with the disease. On November 17th, the Hawaii Department of Agriculture chairperson accepted the recommendation and approved the rule with an effective date of November 20th. And that rule will be published in a number of papers tomorrow in the legal section with the effective date of tomorrow. The interim rule. The interim rule prohibits the intrastate movement of coffee plants and plant parts such as green beans, fruits, leaves, stems, twigs, cuttings, wood, logs, and mulch or green waste, used coffee-related packing materials such as coffee bags, and any previously used equipment used to harvest, transport, or process coffee plants or plant parts, all of which are considered to be potential carriers of the fungus, coffee leaf rust, from the island of Maui, Hawaii Island, or any other island confirmed with coffee leaf rust for up to 1 year from the effective date, which is tomorrow, except by permit issued by Hawaii Department of Agriculture. This is actually fairly similar to what was done for coffee berry borer back when it was discovered. So the interim rule does permit 6 exemptions, which can occur if a permit is obtained from the Hawaii Department of Agriculture. So first exemption is for green coffee beans for roasting or other non-propagated plant parts that are shipped to an approved facility located on an uninfested area that is located at least 2 miles from a commercial coffee growing area or are subjected to an approved treatment. So this would allow the movement of green coffee from one island to another for roasting. For plants, number 2 is for plants from Hawaii Department of Agriculture, certified nurseries, green coffee beans for roasting, or other non-propagated plant parts that are transshipped through an uninfested area of the state for the final destination outside the state. Most often this is going to refer to Oahu, because a lot of stuff doesn't go directly from the neighbor islands to the mainland. For instance, it goes through through Honolulu first and then to the mainland. So this addresses safeguarding that material as it moves onto and out of the island of Oahu. Number 3 is for previously used equipment that is designed to harvest, process, or transport coffee plants or plant parts. Number 4, for plants and plant parts for scientific studies or other diagnostic uses at approved facilities, provided the contents and packing materials used for shipping are subjected to a treatment approved by the plant quarantine branch chief after the contents are unloaded. Number 5, for coffee plants, beans for roasting, other non-propagative plant parts, used coffee bags, and previously used coffee harvesting, processing, or transporting equipment that are shipped between infested islands. So this is actually there's some regulations for moving, for example, between Maui and Hawaii islands. for limited quantities of coffee plants for propagation, number 6, for limited quantities of coffee plants for propagation from an infested area to a non-infested area, subject to a 1-year quarantine in a state-run facility, provided that the Board of Agriculture may reduce that quarantine period on a case-by-case basis. And so what do these permits look like? They're essentially the same kind of thing that many of you have applied for coffee berry borer movement. They're Form PQ7. They're available on the Hawaii Department of Agriculture website under importing and exporting plants and animals, importing plants, and permit applications. And there's a general website address to that. There's also a direct link to that form as well. And the cost is no cost. There's no cost for these permits, so they're free. The applications look, again, they're the same application that's used for coffee berry borer permits, just a 2-page application with several areas to fill out. And with that, I'm going to go ahead and end my presentation, and I'm available to field any questions you may have.
Thank you. Can you hear me okay?
Yeah.
Can you let us know the timeline of when the rule will take effect and when folks can start applying for permits?
Yeah. The rule will take effect tomorrow, once it's published in the papers. And the papers are a number of them: Star Advertiser, West Hawaii, Hilo Tribune, a couple others, both and then Maui, and then Kauai. So it will be published on the 4 major islands. So it'll be effective tomorrow, and you can actually start applying for permits now. The permits cannot be issued until tomorrow. We start applying now. And permits, once the applications are received, they can be they'll be processed very quickly, within a matter of minutes. So we're trying to expedite issuing of those permits. Now, each permit will have particular conditions that address the exceptions, and so they'll be somewhat different for depending on what which of those 6 exceptions you're applying for. But they're all designed to disinfect and then safeguard and track the movement of potentially infested material. So some of the things that are still currently being done for CBB, just with some additions specific to coffee leaf rust.
Okay, we have a couple of questions in the chat. Is DOA exploring any alternate treatments such as freezing, UV, or ozone?
We are looking into those. Right now, the only approved treatment is methyl bromide fumigation, which, of course, there are no facilities on islands to do that. So we are looking into alternate treatments. The conditions on the permit will be broad enough that it will allow us to incorporate new treatments, for example, as they become available, without having to change the permit.
And my understanding is that the permit conditions are very similar to what folks are used to when shipping green coffee for CBB. In other words, double bagging and Grain Pro, with the added step of disinfecting the outside of the bags. Is that correct?
That's correct. And then having the double bags in some sort of additional container, just to prevent the bags from getting torn during transit.
Right. There's a question: is there any threat of spreading CLR in soil?
Soil is already prohibited movement between the islands. There's already regulations in place for that. We did not include that in this interim rule. It seemed that it was a fairly low risk method of moving the rust. And again, there's already regulations in place restricting movement of soil between the islands for other pests and diseases.
Okay, another question. Importing to Kauai includes freezing to control CBB. Are there any studies showing if CLR is killed when frozen that you're aware of?
I'm not aware of any, but again, that's something that as a potential alternate treatment we'll be looking into. The only one, as I mentioned, that we have data on that's been approved is methylbromide fumigation.
Question here, will existing open-ended permits be valid? Do we need to have a new permit application?
Are they referring to coffee berry borer permits?
I believe so, yes.
Yeah, so you will need a new permit for coffee leaf rust because the conditions are somewhat different. For example, you can freeze stuff for coffee berry borer, but right now that's not an approved treatment for coffee leaf rust.
Okay. That answered several questioners. If not, please type again. Can you discuss further the locations where CLR has been found on the east side of Hawaii Island?
I know I can't really give specific locations, but it's been found in the Honolulu area and then in the Honau area. So kind of several locations, I guess, in the Kona side, and we continue to get growers and others sending in pictures that we're analyzing. So we at this point we don't feel we know the complete extent of the infestation on that side of the island.
And how about Ka'u? I saw on your map there was a red dot, looked like an ocean view. Is that a positive identification there?
That'd be a presumptive positive where we saw symptoms, yeah.
Okay, and in Ka'u, are you, or is the department surveying the farms there, or are you relying on identifications from growers?
One thing the department decided early on is that we didn't want to just routinely go from farm to farm looking for coffee leaf rust, because we didn't want to be the source of spreading it from farm to farm. So a lot of our surveys are focused on non-farm feral coffee, residential areas, things like that. And right now we're really relying on the growers to kind of scout their own fields. If they see something, send in photos, and we can go from there. So we're trying not to, as I said, spread it from farm to farm.
Okay, some clarification. The questioner wants to know about the east side of Hawaii in Hilo. What about those?
There's several sites where we found some symptoms and some what looks like spores. We're recollecting and sending samples to USDA to get a confirmation of what we're seeing as being coffee leaf rust. The symptoms don't seem to be as pronounced on the east side as they are on the west side.
And are those presumptive positives, or have they been?
Yeah, they're either based on symptoms or the presence of spores. That's what we're calling the ones on the east side, and until we get some samples identified positively by USDA NIS.
Okay.
At this point, it's been about 4 weeks since we first started looking for it. So we're still in the survey is still evolving. We're still looking, still a lot of places we haven't looked. And so I guess my advice to growers is to even if you're not in one of these areas where we found it right now, is to be vigilant and check your farms and really look for any signs of symptoms and let us know if you do see something.
And is there anything the HCA can do to help with that outreach?
Well, HCA has been very helpful in reaching out to its members as well as the coffee industry in general. I mentioned that the outreach that we engaged in early on, and that a lot of that went through HCA, and even sponsoring weekly meetings on this topic, which have been brought together not only growers and HDOA, but also USDA and other agencies and stakeholders. I think those have been very useful to get everyone on the same page and moving in the same direction. So HCA's outreach and engagement of the industry with all the other agencies that are working on this has been very helpful, I believe.
Another question about your surveys relating to feral coffee. Are you looking at feral coffee on the Big Island?
Yes. Some of the finds there are some presumptive positives have been in feral coffee. It seems to be, especially on Maui, some of the worst infestations seem to be in feral coffee, which you might not be that big of a surprise because feral coffee is not managed at all. It just kind of sits there, and it's seems to grow in areas that are having that sweet spot, as I mentioned earlier, for optimal conditions. So feral coffee seems to be hit harder than a lot of the commercial or farms.
Another question, is it possible to know more details about tentative locations on the Hilo side?
Other than they've been residential or feral, I can't really give more refined locations, I guess.
Can you be general in terms of, it says, question about Hawaiian Paradise Park or Curtistown. Are they that far south?
Can we go back to the presentation and pull up that map? Let's see. It's Kauai. One more. Here we go. So it's basically that area. And again, we're continuing to survey. So if one of these dots isn't in a particular area right now, as the survey continues, we may well find it there. Another thing we're finding on Maui is that it's now started. The rainy season has started, and so some areas that looked negative 3 weeks ago, we're going back to and actually starting to find symptoms now, as the environmental conditions favor growth of the rust. So I hate to say at this point that, for example, on Hawaii Island or Maui, that some areas are completely free of CLR. I would stay vigilant if I was a grower, no matter where you are on the islands.
Next question is something that we've been talking about in our weekly calls, but for the benefit of this audience. Has it been considered to import embryos, vitro plants of hybrids with resistance to several races of CLR for research purposes?
There are ways to do that, and we've been in discussions with the industry and also USDA on ways to do that faster. Because right now, the way the regulations are in place, those plants would have to sit in quarantine for a year before being released. And so we're looking at ways to, for certain situations, to allow that quarantine period to be shortened. One thing we don't want to do is just bring material in that may have, for example, it may not have coffee leaf rust on it because it's resistant, but it could have some other disease or pest on it. So we want to make sure we're not bringing something in that we don't have here that we don't want, in kind of a haste to bring in resistant varieties for coffee leaf rust.
And do we know approximately when it arrived and also the vector upon which it did arrive?
That we do not. Given its spread on both Maui and Big Island, it's probably been here for a while, year or more. As far as how it got here, we don't have any idea at this point.
I think once we identify the race, we may have at least some
We may have
insight.
ideas about. Oh, that's true. I guess I should have mentioned that because we have sent samples to a researcher at the University of Purdue to look at what race characteristics that our coffee leaf rust has in Hawaii. So that those race characteristics give us an idea about geographic area, region of the world where it might have come from. So that might give us ideas to path around. But at this point, we don't know.
Okay. Any other questions from the audience? I will add that working with Kevin has been, he's very effective. He's a great communicator. He's very receptive to the needs of the industry. He's been very responsive. We're very happy to have him in Hawaii working on this.
Thank you, Chris.
Thank you. I got one more. How are public mills recommended to deal with it?
Public mills, like
I guess
a roasting facility?
a toll miller. In other words, you bring your coffee to be milled at a particular location.
Oh, I see. Well, if it's on for example, if you're on the Big Island and you're taking it to a miller on the Big Island, there's no restrictions on that right now because movement intra-island is very difficult to regulate. I think we tried that for the banana disease on the Big Island, didn't work that well. I think we tried it for coffee berry borer, didn't work very well. So we're looking at just restricting island movement between islands rather than on an island. I think if you're bringing your material to a roaster on the same island as you are, I think you should take basic safeguarding precautions. So if your material is infested, you're not spreading it to another location on the same island. So bagging it. Actually, some of the mitigations that are in the permit conditions would be useful for that. Bagging it and disinfecting the outside of the bag.
Yeah, we addressed some of those sanitation protocols at the last session. Andrew Kawabata at UH has a handout which we'll make available, talking about field sanitation, cleaning your equipment, cleaning your truck, cleaning your clothes, et cetera, to minimize the spread. Any other questions from the audience? Okay. Kevin, thank you so much for joining us. We know you're busy. We know you got a lot of work to do. We appreciate your time. We appreciate your responsiveness and willingness to work with the industry to help us manage this. Thank you again.
Thank you, Chris, and thank you for the opportunity to speak to everyone today. And have a good rest of the day, everybody. I'll remain on the call in case there's something that comes up.
Okay, great. Aloha.
Yep.
So our next guest, give me a moment, is Dr. Lisa Keith. Lisa is very familiar to many of us. She's with USDA, which is U.S. Department of Agriculture, ARS, which is the Agriculture Research Service, PBARC. PBARC is the Pacific Basin Agricultural Research Center. She has a bachelor's degree in biology from the State University of New York in Oswego. She has a master's degree in food science and technology from the University of Nebraska. And she has a PhD in plant pathology from also the University of Nebraska. She's currently a research plant pathologist with USDA and PBARC. And from 1998 to 2002, she held the position of a postdoctoral research fellow in the Department of Entomology and Plant Pathology, Oklahoma State University. Lisa's current research focuses on the biology, detection, and management of invasive pathogens causing disease on crops grown under tropical and subtropical conditions. Attempts are made to integrate laboratory, greenhouse, and field-based research to develop and deliver practical disease management approaches that can be adopted by growers. Her main goal is to help solve real-world problems in an environmentally sound and economically viable way. Please join me in welcoming Dr. Lisa Keith. Welcome, Lisa.
Afternoon, everyone. I think we have hit the noon mark. Okay, so sharing my slides. Hopefully, Christian, give me a thumbs up. You see them? Great. Okay. Okay. Well, thanks again, Chris and HCA, for the opportunity to come and share what I guess we know and what we've been learning and really trying to come up to speed on this beast called coffee leaf rust. Today's talk, I really just want to, first and foremost, you hear it, just so thankful for the teamwork, the collaboration, the cooperation from everyone that we're dealing with, and especially you, the growers. So for covering today, it's been a great day of information sharing for sure, starting with Chuck to Andrea to Kevin. You're going to hear a lot of the same things. I figure we need to really understand the problem as best as we can if we want to start dealing with it effectively here. So for today, again, I'll just quickly go over some information on the biology and the disease cycle, looking at symptoms and signs and what those differences are, just the general epidemiology of coffee leaf rust, get into management, and then really focus on what we know and what we don't know and what we're doing here in Hawaii. Okay, so simply stated, for the plant pathologist, well, how is disease caused in plants? And the simple concept of this disease triangle, needing the susceptible host, a pathogen, and the favorable environment. And that seems quite simple, but unfortunately, when it comes to coffee leaf rust, it is very complex. Okay, so you've seen these spores, these pictures, the coffee leaf rust, Hamilia vastatrix, due to just these spiny structures on a good portion of the spore, which help attach to things, and a smooth portion. So as you already heard, these are characteristic coffee leaf rust spores. They really don't look like anything else out there. The important thing is it's obligate. Again, it needs a living host, the coffee plant, to survive. Okay, there's no alternate host found to date, and essentially spores will land and wait until the temperature and the moisture particularly are conducive to germination. Okay, and just to show this again, the spore will land on the underside of the leaf and then germinate and actually go into stomates. And so this is where I want to, and I will give more information on what the difference is on coffee leaf rust, and in particular Cercospora spot, and even others like anthracnose. Okay, this is the big difference. The spore germinates, it goes into stomates, it forms these specialized structures that doesn't disrupt the host cell. It actually just feeds from the host cells. And then as it continues to grow and colonize inside the leaf, it will grow back out of the stomates and start producing spores. And then these spores will go on to more leaves and create more infection. And the process, especially if the weather conditions, the UV conditions, moisture, the leaf health is all conducive to disease, you can have many, many, many disease cycles in one coffee season. Okay. Again, these critical, it's wind and rain, especially wind for long distance transport, rain for short distance transport, but that's the trickiness of this spore. That powdery substance can get on anything and you can easily move it along. Okay, Jacques showed that picture of the powder on his thumb. That goes to somewhere else, and then when it gets to the host and the right conditions, which are the proper amount of moisture, the optimum and range of temperatures for germination and colonization of the leaf, how long it takes to sporulate, how the lesions start to look, and then as they're producing more spores over weeks and then months, and you can see how this epidemic occurs. Okay, now the free spore can survive approximately 6 weeks at the environmental conditions, but it needs the living host tissue to grow and multiply and then cause new disease. Okay, so Jacques showed something like this. Again, you know, you go from this disease triangle, which seems, oh, okay, pretty manageable, to how so many various conditions affect each step of the life cycle of how a spore gets to the tree in the first place, and then how it lands and colonizes and germinates and penetrates, and et cetera, and the circle continues. Okay, the first symptom. So when we talk about symptoms, it's actually how the host is responding to the pathogen. So how does it respond to pathogen attack? The signs are actually the physical presence of the fungus. Okay, so you start to see on the upper surface some chlorosis, again, because what's happening is coffee leaf rust is essentially sucking the life out of the coffee leaf plant. And then the spores start to be produced. And when you zoom in, you can see that you see plentiful spores, okay, this yellow, orange, powdery spore. You continue to see as it expands the chlorotic halo surrounding the lesions, okay? And while the lesions can develop anywhere, due to the moisture, as you already heard today, they tend to be concentrated on the outside of the leaves, okay? As the lesions start to grow and expand, eventually the centers will turn brown or necrotic. The lesions can start to, instead of looking very singular, it's kind of complex and overlap. And ultimately what ends up happening is the leaves will drop prematurely and you'll start to see branches devoid of leaves. Okay, so pictures. I like a lot of pictures, and you're going to see a lot of pictures in the latter part of the talk. Okay, this is the same leaf. Pictures taken by Jacques' group over approximately a 1 to 2 month time period, but you're actually looking at the same leaf. So the initial symptoms on the underside are very tiny chlorotic spots, and then approximately anywhere between 2 to 4 weeks, you'll start to then see spore production. So at this stage, you do not see spore production. It takes amount of time to get in, germinate, colonize, actually now grow back out to produce the structures, to produce spores, and then these lesions, these spots enlarge. So you're actually looking at the same, so this area here, okay, now starts to produce spores. The lesions are getting bigger, and it takes at least a couple months or so to start seeing this necrotic area of these lesions, okay? So again, what you've heard, race is important, level of inoculum, of course. There are more than 50 races. I guess luckily only a few occur regularly. Definitely depends on plant health, how susceptible it is. I mean, you heard all this morning, but there's the reduction in photosynthetic the capacity. That's the damage cause. That's why you start to see chlorosis. The survival depends on the severity of that season, the amount of defoliation, and you can see how this turns into a long-term impact. Okay, Jacques mentioned Lecanicilium. There are known natural enemies. I put up an extensive list, BT, some bacteria and yeast, even some chemical inducers, but unfortunately, it really is not practical for field conditions. So that's definitely an area that needs more testing. Okay, so to change gears as far as what is happening, particularly in Hawaii, and what we know and kind of don't know. Kevin already mentioned, they're doing a great job with the survey work. The points so far, Kauai and Oahu, no detections of coffee leaf rust. This really needs continual survey and monitoring, the chance for early detection and a rapid response in areas that it is not. Okay, it seems to be well established on Maui. And as you're hearing, as the temperatures are changing and harvesting, the incidence and severity levels seem to be increasing. Okay, so there was a question about Hawaii Island, and the positives are these red dots, and I've separated them out. So spores definitely have been seen in these areas of the red dots, but typical symptoms of coffee leaf rust, of what you will see in the literature, HDOA pictures, university pictures, our pictures, these classic symptoms have only been seen in this location as of yet. So this is somewhat of the challenge, that we can find spores, not plentiful, I mean very minimal, but those telltale spores of coffee leaf rust do exist in locations, and this is Manuka State Park, no symptoms, but we found some spores. Okay, so that's something to keep in mind. Okay, so these are pictures from Kona. The typical spores, that kidney bean shape, the spikes all over, that's what you're looking, kind of that gelatinous looking outer layers, actually the spikes that help it attach onto things, and a variety of stages. Now, when you look at these that have a darker yellow and you zoom in, well, you can see there are a lot of spores. It's these initial symptoms that if you're only seeing these little specks devoid of spores, that really can be the challenge for this early detection. Okay, so you kind of have in your head the picture of classic coffee leaf rust symptoms, and I'm going to try to share what is not coffee leaf rust. Well, you can say, okay, Lisa, this is pretty obvious. Things like this are pretty obvious. This, okay, as these are expanding, these lesions, maybe we can see the difference. But, some of these early, wow, that kind of, but that's not it. And so the challenge is, how do you really see it early enough, especially prior to spore development? So here's a comparison side by side. We're talking about the upper leaf surface and the lower leaf surface. And so that's, these spots here are the same spots here. And this is the classic coffee leaf rust, quite advanced. And the severity level, I mean, for this leaf and these leaves are already pretty advanced. Now, you see these spots. Underneath, you see the chlorotic areas, and it doesn't take much to start seeing visible spores, okay? So this is the very early, if you look under some magnification, like a dissecting scope or a hand lens, these very early specks of chlorosis, you can actually start to see spore, and they're the spores, the powdery spores that are now on the surface of the lower leaf, okay? More confusing is Cercospora spot and the early symptoms of Cercospora spot. So again, this is not coffee leaf rust, but you can see how these are very similar to start, okay? When you do look under magnification, you'll start to see a completely different set of fungal structures, a bit erumpent, doing damage to the tissue where coffee leaf rust does not. Remember, it's coming back out of those natural openings. And you also see a depression in the leaf tissue. Okay, so upper and lower surface, zooming in on those newer symptoms of coffee leaf rust, and then what is not. But you can see again how confusing it can be. So I've highlighted the areas, the initial specks, but as you're looking more closely, I'm hoping when you stare at these pictures long enough, you're going to see there is a marked difference, okay? And I've tried to explain these a bit more. So the coffee leaf rust symptoms are small, irregular, pale yellow spots, okay? Approximately 2 to 4 millimeters when we start seeing them. Very small. Initial symptoms on the upper surface of the leaf, but they can be anywhere on the leaf where the stomates are. Again, that's where the fungus germinates and enters into the stomates, okay? They do tend to, you can see how the moisture, the rain tends to develop on the edges. That's really the concentrated effect of the spores now germinating on these edges. All stages of leaf development are susceptible. Again, critical, and what is critically different from coffee leaf rust for other rust, there's no pustule formation. Again, so we're stressing the leaf is intact. You do not see any necrosis to start. You see that slight chlorosis and these spores that are on the surface. And if you actually rubbed your finger, they would easily come off and look powdery on your hands. And really, the mycelium, okay, the other parts of the fungus that are growing are completely within the leaf tissue, okay? This is in contrast to Cercospora spot. The Cercospora spots are actually very circular in comparison to the rust. Very similar size to start, but circular, as opposed to that irregular border that you see with coffee leaf rust. And the Cercospora early symptoms can be on either surface of the leaf, okay? And anywhere on the leaf. So this is one big difference between coffee leaf rust and other fungi, Cercospora, even anthracnose fungi. Cercospora has the capacity to enter stomates, but they also have structures that essentially just penetrate host tissue. So they can directly push their way in to the host tissue, okay? So while all stages of the leaf are susceptible, okay, what ends up differentiating this pathogen from the rust is because of intercellular and intracellular growth of the fungus, it is actually damaging host tissue and results in these erumpent fungal structures and tissue of the plant, and actual depressions due to the collapse of the tissue, so caused by this physical damage from coffee leaf rust and the presence of a toxin. Okay, so early on, even at this stage, you will see tissue damage, this necrotic spots. Okay, so surrounded, circular, there's definitely similarities, but those are the key. And as you can see, you know, the rust only where stomates are. Here's a really good example. It doesn't matter where this fungus, Cercospora, tries to enter. It could be on the vein, it could be, you know, intervenal. It has the capacity essentially just to, like a jackhammer, work its way in. And in no way are the spores of Cercospora or anthracnose powdery where they would come off in your hands. And oftentimes, Cercospora, you'll see concentric rings. Okay, you're not going to see that with the necrosis of coffee leaf rust. Okay, so I thought this was a really good, you know, if I had I should have made a poll or had a test. Coffee leaf rust. So all of these other tiny, particularly in these areas of this new chlorosis, like those 2-3 millimeters, that's actually the rust. But you can see right there, and of course this is a much more expanded lesion of the rust, but right there, while you would see chlorosis, you can actually see necrosis already forming. Therefore, it's Cercospora. Okay, so this slide is complex and a lot of information, but it was a good representation of the types of management strategies for various pathogens and what tools and resources are available. So this was in general for pathogens, but what I did was I kind of color-coded things and made things more specific on what history we all know of coffee leaf rust due to the extensive research over time. Okay, so for the pathogen, the diversity is known, but what I've circled is what is particularly being looked at in Hawaii with researchers here and collaborators. And then these plus signs are things that we do know. So we do know how it's disseminated, coffee leaf rust, and where are potential reservoirs, how they evolve, and new races. I mean, more and more information is being known every day on how these races evolve and become virulent, more or less virulent. What type of host range exists, the types of environmental conditions that is necessary for that disease cycle to occur. But again, circled is what we need to sort out in Hawaii. Well, the diversity of our population and how it responds to these environmental conditions, particularly where these environmental conditions occur and how coffee leaf rust responds in our environment. Okay, so when you start looking at tools and resources, while there are detection methods and resistant plants and control agents and models, these are all things that are and will be done here as well, again, to improve management in our environment. Okay, we have the host and we have the pathogen, but we need to understand our environmental, especially our microclimates. And you can see that there's a variety of things as far as surveillance and quarantine efforts, host resistance, how to eradicate in areas that maybe it's the first detection on an island, how to suppress infections that are already here, how to make improvements in hygiene. Plant health is so critical, and looking at disease forecasting. So there's great resources available and work that can be done here in Hawaii. Okay, so some short-term research that we're involved in, really with the goal is to limit the spread and severity of coffee leaf rust in Hawaii. And that's, again, investigating ecology and epidemiology of the rust fungus, genetically characterize the isolates, develop methods for early detection, develop and deploy management strategies, and then along with the university and HDOA, get information out there as it's happening. Okay, so some work, again, the question was, well, where did it come from? We need to figure that out. It is critical, as far as where what race do we have? And that will give a clue, this population diversity, as we collect more and more isolates to try to figure out where it came from, and was it a single or a multiple introduction over time, and potentially, how long it's been here. So as far as my lab, we do have the capacity. We've been doing some rapid detection with specific primers that detect coffee leaf rust DNA, and then some basic sequencing. So kind of hot off the press, don't quote me because more information's coming, but preliminary evidence suggests similarity to isolates found in Mexico. Okay, you've heard Dr. Amy of Purdue. We are collaborating as far as with her genotyping, understanding the geographical location, actually being able to suggest race. We're hopeful that we're going to get some information within a week or two. And then also working with the experts who do the testing on plants for race identification. And that actually does entail screening a set of coffee germplasm with our isolate to determine specific race. And as we're moving forward, my lab will have the capacity to, so not precisely knowing what race, but in controlled chambers in a lab, we can start looking at the initial testing just to gather some information on the isolates we have and the type of coffee germplasm already growing and how susceptible it will be, particularly under the best environmental conditions. Okay. If you've been on meetings, we mentioned the opportunity of using spore trapping potentially as an early detection method. So I wanted to touch upon that just briefly. We use this equipment called roto rods, and these are actually two separate roto rods. What we were doing for actually another project is trying to determine, well, did it matter which way the rods were facing? In this case, the rods are actually up, and in this case, the rods are actually down, and it's a battery powered, so we can connect to this battery here, and it's programmable, and you can time it to have these small rods that are actually coated in silicone grease. Once this starts spinning around, the rods are now going through the air and trapping anything that's in the air. Okay, and what's very interesting is that we can program these to be triggered by wind speed or a change in wind direction. So you can see the thinking of potentially utilizing this as an early detection method. And then we have the capacity to actually take these tiny, this is blown up, but maybe they're like an inch long rod, and actually use them for the rust identification. Okay. So I had mentioned for our rod project, we had been putting out and doing a study to see if we could find the spores of the rod fungi, which once it really reached a certain level, we could find the DNA all over. So that was not really worth having ongoing research on Hawaii Island. But for Maui, we had placed 3 roto rods where our partners, thanks to DLNR and MISC, have been looking after and sending us roto rods, and also one on Molokai. So with the 3 locations on Maui, usually it was going on, this started about a year ago. Monthly we've been receiving, and so what we did, we pulled those back out and actually used the coffee leaf rust specific primers to see if we could pick them up. And I guess in a way, good to say, we haven't detected coffee leaf rust spores in any of the traps. And I mention a passive trap because that was really just really like a microscope slide with some silicone grease placed out in the environment. And if something landed on the slide, we could try to detect it, versus actively, moving through the air to collect and trap what is blowing through the air. So I guess in a way, I'm hopeful maybe that's a good news sign we haven't detected. So, the limits of detection, we haven't really gotten to that to say, wow, it is so plentiful and blowing through the air. Okay. So I would just like to, I'm coming to the end. Just mention, some of these other things that researchers at ARS in particular are going to try to tackle. And, specific questions. So there are names addressed with research. This is more short, medium, long-term research heading into longer-term research. But if you're interested in this type of work, please contact the name. So Dr. Melissa Johnson has been very involved in the monitoring efforts of CBB and really feels confident that those methods and these weather stations and picking up data for management and weather can actually be easily applied to collecting this sort of data to understand how this will influence coffee leaf rust incidence and severity. Okay, so they've been collecting for a number of years at a variety of farms in Kona, Ka'u, and Hilo, of various elevational ranges, and they collect temperature, relative humidity, rainfall, wind speed. These sensors have been in place and are continued to be in place because of CBB. So the addition of leaf wetness will, these sensors will actually then complement all of these environmental conditions and the standing leaf moisture, which would be necessary for germination to take place. As we move along, the management strategies, what's working, what's not working, the types of sanitation, that's an exciting prospect to really better understand all of these microclimates and when to trigger management. Another aspect is actually pre-visual detection, utilizing remote sensing technology, again, because the health of the plant is so critical. This really, along with ground truthing, can become a critical component of the research. Okay, so Dr. Roxana Myers, our nematologist, is actually currently working on some research and has some future plans to evaluate coffee leaf rust resistant coffee varieties for tolerance or resistance to coffee root knot nematode. She's currently doing some potted plant bioassays, screening some of the more resistant varieties of Obata and Tupi, and then for future, actually doing field evaluation of resistant varieties grafted onto tolerant rootstocks and screening the resistant varieties from the World Coffee Research for susceptibility to nematodes, because obviously Kona suffers, from that major issue as well. And Dr. Tracy Matsumoto, along with Dr. Chifumi Nagai at HARC, are actually participating. So we are participating in the Coffee World Research, an international multi-location variety trial. I was like, whoa, where are we? Well, we're right there. We're represented right there. And so more information, definitely talk to Dr. Tracy and Dr. Shifumi. And okay, so I know I've kind of taken my allotted time and then some, but really I'm the optimist. Okay, I always want to think, hey, we got a shot. The coffee leaf rust has been around for 140 years. Okay, so it is well studied and well managed in other parts of the world. What's really critical for us in Hawaii is figuring out what race it is, having a better understanding of incidence and severity here, and what is going to work best in our environment. Okay, you've heard time and time again today, and you will continue to hear healthy plants. That's a major role in combating this pathogen. We really need to eliminate inoculum and eliminate it early if possible, really trying to minimize the spread of coffee leaf rust. And I've addressed, and it's not just us, but ARS and others really have a plan moving forward with short-term and long-term research strategies that, again, as we learn more, we will distribute and dispense to all of the growers and work closely with growers as we're doing the research. I mean, that's where we're really thankful for such good collaboration. And with that, I will say thank you very much for your time, and I'm happy to answer any questions.
Thank you, Lisa. That was very thorough. A lot of information there. There's not a lot of questions, but there are 6 people watching. One question outstanding is, who's the manufacturer of the rotor rod?
That's a good question. I'll have to get back to you. I can add that into your chat. We actually ordered them from Canada, so that was a little challenging to start. We do have more available in our laboratory to utilize right away. But that's so funny. I will either, as I'm doing things, I'll try to get that info right now, but I will definitely get the info out.
Okay, thanks. I did Google it while you were speaking, and
Okay,
I found
good.
some information. I haven't gone through all of it. So if you Google rotor rod, you'll find quite a bit of information. Question, how will it help to know the race of CLR?
Well, so race to genotype, really when it comes to coffee leaf rust and coffee, there's a lot of gene for gene interaction. And you heard Jacques mention, the more widespread and aggressive race 2, we really need to understand what race we're dealing with, which then gives us a better understanding of the susceptibility of the germplasm that we grow currently and hope to grow in the future. So you really need that race determination to understand what you're working with, how aggressive it is, and how the host is going to respond to what's here. And I think it's also important, you really need to find that baseline of what is here and constantly look to make sure there are not future introductions, okay, additional races coming in. But as Jacques mentioned, these fungi can now change. So they have the capacity to start, there's 50 plus known races. And as you heard, many currently unidentified. And so we really need to see what is here and how that population kind of becomes in the future.
Okay, thank you. Next question. If we don't have it yet, best practices for prevention at a farm level.
Yeah, so Andrea definitely gave a lot of really good information. And it is what you're hearing as far as plant health it is surveying. I mean, that's what is available now as far as fungicides are protectants. I mean, that's really going to be the biggest challenge, in my opinion, combating this disease. Everywhere else in the world, depending on the incidence level, approximately 5%, 10%, you have to work in systemics, and we do not have that available as of yet. And that will take some time. I mean, people are working on that, but it's going to take some time. So catching it early for the capacity of using protectant sprays really is critical. So, you know, Andrew went into that. It's the pruning, it's the health, it's the constant monitoring, particularly the lower portion of the trees, really looking. But do keep it in mind, you know, as you're looking, you really should practice that hand sanitation between leaves, well, trees for sure. I mean, you're looking at your tree. Well, if it's those initial symptoms where, well, you don't think you see spores yet, but you can easily pick up the spores. And now you go looking on other leaves, you can see why people could easily move it. So to avoid that with your own farms, definitely have that sanitation. You know, it's like practice the COVID after every tree, using that hand sanitizer. And as Andrea mentioned, you know, the coppers in particular, they are beneficial against Cercospora and anthracnose and coffee leaf rust. So kind of the combination there.
Another question, follow up in that vein. Would spraying a product like Surround prevent infection?
You know, I haven't come across a whole lot of that in literature. If just based on the protectant, if it were there prior to something landing on the leaf, that may offer some. But I'll be honest, that really scouring the literature, that is not a major form of management. I mean, unfortunately, that was that list of biologicals and, you know, other disease response. You can put some things on that actually trigger plant defense. Nothing is being utilized at a scale, you know, the farm scale level. So there's potential, but not part of an overall global management strategy.
Okay, I think we know the answer to this one, but I'll ask it just the same. If every coffee farmer on Hawaii Island eliminates coffee leaf rust, won't feral coffee continue to spread it?
Yeah, that's a challenge. Okay, so here's my educated response, guess to that. It will continue to spread it, to serve as a source of inoculum until the tree actually dies. Unmanaged trees, I mean, with zero management, trees will not live forever with coffee leaf rust. Okay, so it's going to be a matter of time. Enough defoliation will occur. Minimal leaves will be produced again. The tree will die. So that, but it's not going to be an immediate effect. I mean, you're going to have seasons, potentially years of a source. But that's kind of it's not going to be a forever source. But again, that's the management has to happen. Really at the earliest possible time.
Another question. Will the burning of contaminated coffee materials release spores into the air?
That's a really good question.
I'm wondering if better to compost or burn drop leaves or trimmings.
Okay, so that's a really good question. Oftentimes, the management is spraying fungicide, so actually killing the fungus first prior to burning. Now, I came across a paper or two that some fungi can actually be caught up in the smoke as the initial burning has happened. Now, to the best of my knowledge, I have not read that for coffee leaf rust. So in theory, it really should be devitalized with those high temperatures. And the same thing then with composting. You have the temperature ranges for what the fungus can survive, the optimums for when it best produces disease symptoms. There's been a lot of questions as far as, well, cold tolerance or heat tolerance, and that is true that they're more sensitive to high temperatures than they are to lower temperatures. And there are studies showing that spores can be frozen at -20 or -80 and not lose 100% viability, if that makes sense. So even those extremely cold temperatures, they will not kill it 100%. However, the risk of a new infection really comes down to having the viable spore land on that coffee plant with the right temperature and the right moisture and everything else going on to germinate and start a new infection. So I think that's more of the rate limiting step, if that makes sense.
Yes. Do you have any recommendations for handling disposal of infected leaves and plants after stumping? Kind of in that same vein. So
yeah,
what do you recommend?
that's the challenges. You don't want to take that to green waste. You don't want to, it's like, okay, mainland, bury it, try to bury it. Putting, you can really, anything to help heating up the material. You potentially could put tarps over the material, which limits, aerial dispersal. It'll start to heat it up. The plant material will die. There is no living host for the spores. So it's really, there's research where herbiciding to kill plant material, kind of in combination. The sprays, definitely spraying contact fungicides. And that's really, I think, the best we have at the moment. And you also have my contact info, so I'm happy to, at any time, I'm here to answer questions.
Okay. Well, we'll make that information available. The session is being recorded. The presentations will be available on our website, as will the recordings. One last question. Does the 165F rule apply? I'm assuming that means 165 degrees Fahrenheit.
Oh yeah, that will devitalize, but I'm not sure if that's what the question is.
Yeah, I think it is. May have to get back to him on that.
Well, I appreciate it very much. And again, we really can't thank you as growers enough for just the cooperation and collaboration. Really appreciate it.
Thank you, Lisa. We appreciate all your great work and clear communication, making yourself available, and likewise for your colleagues. Thank you so much. Aloha.